So what I'm going to talk about is I'm going to take you through some of the work we've been doing, which has essentially been working around the principle of is Parkinson's disease one disorder? Is it several disorders? If it's one disorder, does it always follow the same path, which I think we know the answer to? And then using that information, how can we understand why people behave differently with Parkinson's disease? And then ultimately, what impact that will actually have on therapeutics. So I'm going to start off by talking through a epidemiological study, collecting people in as it was in the county of Cambridgeshire over a period of time, what we found when we followed these people, how that's been used to make models, and then how we're starting to understand that in the laboratory, and then I'll end up on therapeutic. So you have to go back to the late 90s, which is when we started this work, which was very much the view. Nowadays, people have a much more sophisticated view of Parkinson's, but when we started this work, there was a general conception that really Parkinson's disease was primarily a movement disorder, which is true. It's primarily a pathology of the dopaminergic system, which is true, and that's pretty much it. But it was quite obvious that this is not the case, that it's much more than this. And so what we decided to do back in 2000, Tom Falterney, who was the clinical fellow at the time, he's now a professor with Hugh at Queen's Square, we set about trying to collect people with Parkinson's disease and follow them over time. And the hypothesis was something will appear out of the data in time that will be interesting. So we didn't have a theory that there would be 3 types of Parkinson's disease, 5 types of Parkinson's disease, 1 type of Parkinson's disease. We thought, why don't we just collect everybody in the county of Cambridgeshire over a 2-year period and just follow them over time and see what happens. And this has proved to be quite a successful project. Not just because we can publish a lot from it, but it's actually told us quite a lot, which is instructive. And amazingly, this project, this study, is the only, at the time, the only study of community-based incident Parkinson's disease in the world in the post-levodopa era. So all of the statistics that people say about the average age get it, mortality, everything else, was based on samples which weren't based on what actually happens in real life out in the community, as opposed to what happens in clinics, in hospitals or specialist centres or whatever. So this was the study. So we collected everybody who could conceivably have Parkinson's disease over a 2-year period in the county of Cambridgeshire, and we have followed these people over time, and we can continue to follow them to the present day. And so we essentially said to the GP, send us anybody you like, anybody you think could conceivably have Parkinson's disease. So we saw people with arthritic shoulders, we saw someone who had muscle aching in their arm, we saw all sorts of people, but we did see people with Parkinson's disease, and we characterised them at baseline, and then we followed them up, and we're currently just finishing the 15-year follow-up data on this cohort of patients, which is called the CAMPAIGN study. And essentially, this gives you some of the statistics on it. It's not particularly easy to read, but I think there are a few interesting things to take away from it. The first thing is that the average age of Parkinson's disease in the community, so this isn't in specialist centres, in community, is actually 72. Whereas in America, they'll tell you the average age of Parkinson's disease is 59. So in America, it's the average age of Parkinson's disease that you get into healthcare systems in the States. And that makes quite a big difference, actually, when you're trying to think of health economics and planning for treatments. And essentially, we found that the incidence increased with age, as you would expect. And the other thing we found was that about a third of patients, a quarter to a third of patients at presentation had some abnormalities in thinking or differences in thinking. And that we've pursued, and I'm not really going to talk about that, but the interesting thing about thinking differences in Parkinson's disease is that not all thinking differences are a harbinger of bad things. So I think there are 2 major problems with thinking in Parkinson's disease. One relates to dopamine, and paradoxically gets better over time, and the other is predictive of the more long-term complications, such as the dementia, which I'll come on to talk about in a minute, because that was one of the main measures we used by which to try and get to what we think goes wrong in bad Parkinson's disease. So that was the CAMPAIGN study. Started it at the beginning of the century, collected all the information, and then in order to validate it, we thought we would do the same again, which we thought would be very easy. So we simply collect people over a longer period of time in the same area using the same process. Well, that proved actually not to be very easy because regulation had changed at that point, so we couldn't actually collect information in the same way from hospitals with discharge summaries. In the original study, we'd offered GPs £10 and a quick outpatient appointment for patients who were referred to our study. That was deemed to be unethical, so we were not allowed to offer GPs any money or any preferential treatment of people who entered into the study because it was unfair on those who decided not to join the study. So all of these things added a level of complication with it, but nevertheless we collected all of the patients, and when we actually compared the epidemiology between it, they were pretty similar. The mean age now was 69, but essentially the statistics were the same. And this has become important because obviously when you do one study, it could just be that's the way it is in Cambridgeshire in 2000, 2001. It's not like it is anywhere else in the world. And of course, we haven't looked anywhere else in the world. But using the same sort of methodology over a 5-year period, we found essentially the same group of patients. So it would probably be a fair reflection that that is what Parkinson's looks like in the real world out there in the community. And the other important point is it increases the number of patients, which allows us to follow people for longer periods of time with bigger numbers, which allows us to have more predictive value than some of the things we're trying to prove. So we've now, as I say, collected this data, and what have we found? So the first thing is, which is a bit gruesome, if you like, or you might not be want to know about it, but the death rate. So the mortality rate. So if you have Parkinson's disease, does it increase your death rate? The answer is no. So if you look in our cohort of people we collected in Cambridgeshire and we compared them to an age and sex matched population in Cambridgeshire, if you had Parkinson's, it did not shorten your life in any way at all. So the rate at which you died was exactly the same whether you had Parkinson's or not. If you look at death certificates, only 1 in 10 death certificates had Parkinson's disease on it at all. And so the vast majority of people with Parkinson's disease actually die with it, not of it. And that is important, again, for health economic reasons. So when you're trying to justify treatment, you have to try and work out what's the best way you're going to target treatment. So lots of crackerjack treatments, which we talk about, will not be suitable for everybody. So the vast majority of people with Parkinson's are going to be fine with standard treatment. However, there is clearly a group of patients, younger patients, where the treatments that we have at the moment are just not adequate and we need to do something better. So that's what we found with the mortality rate. But the thing we were really interested in was looking at cognition, because what worries people with Parkinson's, and you would know this better than me, is what's going to happen to your thinking. People worry about the motor aspects of it, which we can manage reasonably well, but people worry about the thinking aspects of it. And so it is true to say that if you have Parkinson's disease, it does increase your risk of having dementia overall, 2.5-fold increased. So then that raised the question, well, that increases the risk, but clearly not everyone develops this, and also not everyone follows the same path across the disease. So then what we did was we plotted the data. So then we said, what happens over time? So this is simply, you collect everyone in an area, you diagnose them with Parkinson's and say, I'm just going to follow you and see what happens. So what you find is, these are a number of measures here. So at about 10 years, which is where this data is, about 50% of people with Parkinson's are still alive after 10 years. Well, average age is 72, as I say, so average age is 82. So that, as I say, is exactly what you'd expect for that age of population. In terms of falls, so this is postural instability, a thing called Hearn and Yahr stage 3. This is a major milestone in the landscape of Parkinson's disease. At 10 years, about 2/3 of people have falls. 1/3 of people do not. I'll come back to that. If you look at the dementia, which I was talking about, 40% of patients, by the time they've had the disease for 10 years, have developed a dementia. But 1 in 5 patients, so 20% of people with Parkinson's disease, 10 years into the illness, are still alive, have no falls, no major thinking problems. So they're doing really well. So that, of course, then begs the question, why do they do really well And why do they do really badly? Because they've both got Parkinson's disease, but clearly they have behaved very differently. So we set about looking at this particular group of patients to try and understand why it was that these patients did less well, and conversely, why the other patients have done well. Now, the beauty of this study is, of course, when we collected all the information in 2000, we had no idea where people were going to go. So we can now look at all the people who have developed a dementia, who didn't have it when they came into a study, and say, when we saw them right at the beginning, what were the things which we measured which were predictive of where they were going to go? And so that's what we did. So we looked at these predictive measures, and this will now distract you because you will all spend the time doing these cognitive tests to see exactly where you are. So we went and we looked at the baseline measures. So we did tons of computer testing. We did all sorts of sophisticated testing, but fundamentally, and this is what I do in clinic now, you can predict within about 3 minutes, 4 minutes of someone coming into clinic with newly diagnosed Pugsies, what their chances are of doing well or badly. And that relates to age. So actually, if you're over 72, so you're over the average age when you get it, that is the biggest prognostic indicator of doing badly with Pugsies. So age is an incredibly important factor, and I'll come back to that because when we come to try and model the disease to understand what goes wrong, a lot of the strategies which are currently being developed, I say, fail or are likely to fail because they do not take into account age, which is the biggest dominant factor for all of these types of diseases of the brain, including Parkinson's disease. But when we looked at the original data, there were 2 tests which were highly specific for predicting people doing badly. One was a thing called semantic fluency, which was getting people to name as many animals as possible in 90 seconds. The standard test is doing it in 60 seconds. I have absolutely no idea why we did 90 seconds. No one knows why we did it for 90 seconds, but 90 seconds is what we did. And if you could not name more than 20 animals in 90 seconds, that was a bad prognostic sign. So if you're over 72 and you can't do more than 20 animals, that's not good. And then the other test, which was highly predictive, and I personally think this is the most predictive test in clinic because it's so easy to do, is copying this interlocking pentagon. So if any of you come and see me in clinic, this is I'll do it in everybody, I'm not just picking on people to do this, but people who are going to do badly with Parkinson's disease really struggle with this, and the thing they really struggle with is seeing that it's an interlocking pentagon. So those lines there become a complete mystery to them, and they end up drawing it in different ways to indicate that they haven't understood what that actually looks like or what that figure's meant to represent. And that's highly predictive. So if you're over 72 and you can't do 20 animals and you can't do that, your chances of having a dementia in 5 years is about 80-fold greater than if you're under 72 and you can do both of those. And I would actually say that this is not predictive of early dementia. This is the very first features of it. So we are picking people up at the very earliest stages. And that's going to be important, relating back to a point that Hugh just made about picking up people early in the disease course to implement new treatment. Because if that is true, then that's when we should be treating people with disease-modifying therapies. At the moment, they present not within the first 3 years, not 5 years, not 10, not 15, but through the door. And I'll come back to that at the end. So those are clinical measures. So now, based on this epidemiological study, we can see people with Pugsies and we can predict when they come in whether they're going to do well or do badly. That's fine, but the question is, well, why? Why do some do badly and some do well? Why can some people not do this and some people do it very well? Well, obviously, the younger you are, the easier it is. So age is a factor. I don't know what age is, but that's an important factor. The next thing we did was we went looking for genes. And Hugh may have mentioned this, and I apologise, but we went looking for genes, not genes that increase your risk of getting Parkinson's disease, which both of the genes I'm going to tell you do, but whether they change what happens if you get it. And so there were 2 genes that we looked at, because they happened to be genes that were, first one was sort of a gene very close to Cambridge because they've done most of the work on it. And this is a gene called the tau gene. And this increases your risk of getting Parkinson's. So Hugh was the first person to show that the H1 tau haplotype, which a particular collection genes, increases your risk of Parkinson's disease. Not a huge amount, but it increases it. So we then looked at our population to say, well, I'm not interested whether it increases your risk. If you've got it, does it change what happens? And the answer was yes. So if you possess this H1 tau haplotype, which is what the majority of us have, then actually you do rather badly with your Parkinson's. So this, whatever this is doing, is accelerating the disease process. So in some way it's contributing to what's going wrong in Parkinson's, or conversely, if you're the so-called H2 carrier, it protects you. So that now gives you a bit of a clue that there is some genetic variability which is not only influencing whether you get Parkinson's, but the course you're going to follow. So in somehow that's driving the disease process. So that was one gene we discovered. There were lots of genes we discovered had nothing to do with it. So it wasn't just every gene we looked at we found something. The other one was this gene, which in 2000 was absolutely nowhere on the map with respect to Parkinson's disease. This is a gene associated with Gaucher's disease. You're probably aware of the fact if you inherit 2 mutations in this gene, you get Gaucher's disease. If you inherit 1, it increases your risk of Parkinson's disease, and in fact is the commonest genetic risk factor for Parkinson's. So in our population, when we looked at all of our incident cases for the genes which are commonly associated with Parkinson's, LRRK2, well, that's really the only common one, but you know, the alpha-synuclein pink, all of those. Of all the incident cases, we now have 2 cohorts, we found none. Didn't find a single LRRK2, didn't find a single patient with one of those. 10% of those incident patients had a GBA mutation or something that could be a GBA mutation. So it's common. So 1 in 10 people have it. And that will influence where we go forward with drug trials, because you can enrich for this population given how common it is. So this increases your risk of getting Parkinson's. But what else? What happens if you have Parkinson's disease and you have this gene? Well, in our cohort, if you actually had a single mutation, you did really badly. So in point of fact, within the course of our trial, of our study, every single person that had a mutation who presented with Parkinson's disease ended up developing a dementia within 10 years. So these 2 genes not only influence whether you get Parkinson's disease, but where you're going to go with it. And that gives us clues, as I say, into what may be driving the disease process. So this has implications. So we could, coming back to the question I think you asked at of a few, we could in the future be seeing people with Parkinson's, not predictive testing, you know, do I do a LRRK2 mutation test on someone from a family? You could say, I see someone with Parkinson's, I do my clinical measures, I do selective genetic testing for genes which I think are going to influence the disease course, and then that puts you into a certain category which may influence where we go with our treatments. So treatments which work on GBA or glucocerebrosidase, you know, you may want to have this test so we can enrich for people with that. Now, you can use this information to try and work out what's going wrong in Parkinson's, and I'll come on to that in a minute. You can also use all this information to start building models. So you can use this to predict where people will go. So this has 2 implications. One is you can see someone in clinic and you can now start to say, I think I know where you'll be in 5 years. I think I know where you'll be in 10 years. I think I know where you'll be in 15 years. Now, obviously, these are very early days, so we don't know how reliable this is. The other advantage is it changes how we do clinical trials, because you could now say, well, I'm going to take a bunch of people with Parkinson's disease, shove them into my model, and I know, for example, let's take 80-year-old patients, can't do semantic fluency, can't do pentagon copying, tau mutation. We could predict, say, well, in 5 years, 12 of 20 patients will be demented. I know that because that's what I've predicted. I'm only going to take 20 patients of that time, and I'm going to give them my drug. I'm going to wait 5 years, and if none of them have demented, well, that's the end. I mean, it's solved. So we don't need studies of thousands of people. If we can predict accurately where people are going to go, and they don't go there, then we may have actually found a better way of doing trials without control arms and all of that issues. I'm not saying it replaces it, but it's a new way of thinking about these type of trials. And so we've used it to model where we think people will go. So in terms of the community, there's a big group led out of Harvard by Clemens Scherzer, who looked at all of the Gaucher patients that had been collected, the GBA positive patients. So we provided our cohort, he provided his cohort. I think there were 9 in this, and they've used that data to predict where people will go with a GBA mutation. And Caroline Williams-Gray, who works with me and runs her own group, did exactly the same with a group in Holland and came up with a formula which allows you to predict where people will be going. It looks very complicated, but actually it's quite simple, really, because it's just age. It's your Unified Parkinson's Disease Rating Scale on the axial bits, the mid bits, the semantic fluency, which we did it with the Dutch, so we had to correct for language, because the different numbers of animals you can produce in 9 seconds, depending on what language you are, with these correction factors. And we validated it because we did it in our study, because we knew where patients were going. So we said, here's the baseline data. We'll model where they're going to be in 5-10 years' time. And the Dutch group had done exactly the same with their cohort. So they then said, here's our model. We've predicted what will happen. Test it on your cohort. And it proved to be fairly accurate. So now we can start to actually model the disease, tell people where they're going to go, and it will have an impact on clinical trials. Now, the other thing is to try and work out what this means for the disease process, which will not only tell us why patients do it biologically, but what can we do to help them. So you could imagine tau, for example, is involved with how nerve cells communicate in a thing called axonal transport. So it tends to shove things down the nerve cell, collects things at one end, and keeps the nerve cell happy. You could imagine that what the tau haplotype does, nothing major, because most people don't get it to 70, so it works pretty well, but you could say, well, it actually reduces a 4-lane motorway to a 3-lane motorway. So as time gets on, and one of the lanes starts to break down, you can start to see that axonal transport shifting good things up and down a nerve cell is marginally impaired. So that makes the nerve cell a bit unhappy, and when it starts to go wrong, it makes it even unhappier, so the disease starts to speed up. The GBA, which is that Gaucher's gene, which I was telling you about, probably has something to do with clearing rubbish out the cell. So it's a thing called the lysosome. The lysosome is like your dustbin collection lorry. So it basically just ships out all the rubbish which is in your cell. Well, it may be that GBA mutation just says, well, it doesn't work quite as effectively. So whilst you would normally be clearing out your cell once a week, when you have this mutation, it can only do it about once every 10 days. It's not working quite so well. So now you've got a rubbish collection that's not working as efficiently. You've got a transport system that isn't working quite so efficiently. Chuck in a bit of age, and then things start to go wrong. And then when it goes wrong, it accelerates away. So can you study that? I mean, that's a great theory, but can you actually do that? Well, we haven't just. We've been doing this for years, but we've never got it to work. But we just started to get it to work now. So what we've been trying to do is we've been trying to study this in the patients who we have predicted where they're going to go. And the way we do this is we take a bit of skin cell, so whip a bit of skin out of somebody, grow up the skin cells, which are called the fibroblasts, and then most people, when they go that, they then reprogram them into these things called induced pluripotent stem cells, which many of you have heard about, very trendy, sexy type things. And then you turn those back into dopamine cells, and then you can study patient cells in the dish. It's a great idea, and lots of people do that, but what we do is we grow up the fibroblasts and then we directly turn them into nerve cells. So we don't take them back through pluripotent stem cells. And the reason for that is that the one thing you do with absolute certainty every time you do this pathway is you take your average 72-year-old fibroblast and you turn it into a minus 6-month stem cell. So you've put it back to the very origins of life, which of course is why they're very exciting, but you've lost the very thing which is critical for getting the disease in the first place, which is age. Now, I don't know what age is, as I keep saying, but these cells, these induced neurons, have all of the characteristics of age-related nerve cells. So these are old nerve cells. Now, this system, to try and make these adult nerve cells from skin cells, proved to be a bit of a labour of love. I'm very fortunate that I have some fantastic collaborators in Sweden who managed to get this to work. So we could make nerve cells from skin cells from mice, dead easy. Old mice, dead easy. Fetal material, humans, dead easy. Adult human, rubbish. So the one population we wanted, we couldn't do it. Anyway, this, which I will test you on later, is the cocktail that we came up with, which now allows us to actually take skin cells, dry them up in the lab for a few months, then you apply these factors, and then you start to see these little nerve cells emerging after a few weeks in the dish. So this is a patient-derived nerve cell which is old. And now we can start to study what's different between our different types of patients in the dish using this particular approach. Now this will be very helpful, but this will tell us what goes wrong in nerve cells. And the assumption has to be that in Parkinson's disease, everything that goes wrong in your nerve cells. That may be right, but there are other players. So in your brain, you have lots of non-nerve cells, so glial cells. So are they important? They just support the brain. Einstein was said to have 40% more glial cells than the average person. That has been used by glial biologists to say that's why you should study glial cells, because it clearly makes you seriously smart. But glial cells clearly have a role to play. The other cells, which we think are fundamental to what goes wrong in Parkinson's disease, is actually inflammation, your immune system. Now, why do we think your immune system in some way is important to what goes wrong in Parkinson's disease? Well, many years ago, we did a study where we looked at genes which control your immune system. So this is the HLA. So this is a gene which is associated with all autoimmune diseases. So multiple sclerosis, which is an autoimmune inflammatory disease of the brain, HLA association. Thyroid disease, Graves' disease, HLA association. Rheumatoid arthritis. All of these diseases which have an autoimmune basis have a linkage to HLA. Now, in Cambridge, we have tons of people who study MS. We had all of our samples from PD, so we thought, let's just have a look at it. And we found an association. So we thought HLA is associated with Parkinson's, so perhaps the immune system does have something to do with it. This was then followed up a year later in a much bigger study, which I'm not bitter about, but that one's all cited, and this one never is. But that was basically proving or supporting what we said. Caroline in our group then went back and looked at samples we'd collected of all of our patients when they first presented with their Parkinson's disease to look for markers of inflammation. So she went back and looked at these newly diagnosed patients. We took blood from them. We measured in their blood at the time they came in, measures of inflammation, followed them over time, and looked at what happened over a 3-year period in this study. And what we found was that people in a blood sample that had a profile that was of pro-inflammatory, so things that were causing increased inflammation, patients did less well. So they ended up going quicker to the stage when they were falling and developing major thinking problems. And conversely, people who had a peripheral immune profile, so it's in your blood, not in your brain, which was anti-inflammatory, so the sort of profile you get when you're producing a response which is damping down inflammation, did much better. So now we have a peripheral marker of inflammation that seems to, in some way, be driving the disease, in the same way as we've already now found markers within nerve cells that seem to be driving the disease. We then have taken this to the next stage, which we have yet to write up and publish, which was that in our population of people we followed over time, many people are very generous and donate their brains at an appropriate time during the course of their illness. And when we look at those, when we look at those, we have found something which has supported this. The other piece of information I just forgot about was actually, interestingly, when we looked at those campaign patients, the ones I told you right at the beginning, and we just said, which patients were taking anti-inflammatory drugs at the onset? So these were people who were taking drugs for arthritis, aspirin for heart disease and stroke. So it wasn't a properly controlled study, but interestingly, people who were taking drugs which dampened down their immune system for other reasons actually were protected at least for 5 years from developing a dementia. Now, it could be there are all sorts of reasons for that, but again, it's another piece of evidence that inflammation may be important in Parkinson's disease. And the final thing we've done is we've been collecting brains from patients as part of this study and looking at markers in their brain. And what we have found is that the most positive finding in our brain samples predictive of where people are going is not how much of the abnormal proteins they have in their brain, but how much inflammation they have in their brain, which has led us to propose that what might go wrong in Parkinson's disease is that there is something wrong inside cells which starts the disease process. As those cells start to die, they produce an immune response, which may be out in the bloodstream. Those immune cells get into the brain, activate brain immune cells, and drive the disease process. So if that is true, there are then essentially two major ways you could target it just based on what I've told you. One is to try and get into the cells themselves and stop what's going on. The other is to try and target the immune activation and inflammation, which seems to be important in driving the disease process. Now that's highly tractable because we have hundreds of drugs which can target the immune system. So, not quite sure of the time, I'm probably banging on for too long. So I'm now just going to quickly sort of take us through what does this mean therapeutically. So what's the value in knowing all this? So some people always tell me, this is seriously depressing, Roger. Do I really want to go and see you? And you sit there and do a few tests and say, you're going to do badly. Of course, I don't want to tell people they want to do badly, but that's the reality. So we have to at least engage with it and try and understand that we can at least predict where people are going, which has advantages. But it also has, not only in terms of understanding the biology and therapeutics, it also enables us to start thinking about therapies. And that's important not only in the future, but also helps explain the past. Because, as some of you have heard me say before, if you do not take this into account, you will start to end up in a mess. So, for example, in an area which I'm particularly passionate about, which is dopamine cell therapy or gene therapy to restore dopamine cells, patients over here are going to do very well with dopamine because they have a relatively benign disease, they progress slowly, all the problems are really motor. These patients over here sure have dopamine and motor problems, but they have a much more malignant disease. This goes everywhere, this causes thinking problems, this causes falls. Now I start a new experimental therapy, best cell therapy in the world for dopamine. I take 10 patients at random. I want to be nice and generous, so I want everyone to come in. I take 5 from here, 5 from here. Now, you tell me what I want. I don't want a motor measure, I want quality of life. So I'll say, all right, I'm going to wait 3 years. I'm going to ask you, do you feel better for the therapy? So this group, crackerjack, terrific, love your cells, did really well. This group, cells have done equally well, but the problem here is they have thinking problems, psychiatric problems, falls, nothing to do with dopamine. 3 years later, do you feel better? You say, no, I'm not better. In fact, I'm much worse. So much worse, much better, sum them together, no difference, therapy in the bin. Let's try someone else. So it has important implications. So we and others are starting to think about how we can use this information to inform us about trials. So on this side, we would say this group of patients, which I suspect is the majority in this room, will be the type of person who will be best suited for anything that targets the dopamine system, whether it be cells or genes. This group, who we traditionally exclude from all trials, are the ideal group for disease-modifying therapies because we know they're going to do badly, they're going to progress quickly, and we're going to find out problems and solutions much quicker by using that group. So where have we got to with these therapies? This is slightly outside my brief, but I'll just tell you. So on that group of patients, we've just finished treating 11 patients with fetal dopamine cell transplants. That was the last patient was grafted 2 weeks ago in Sweden. And we're now moving towards stem cell derived trials. With the UK, I imagine it will be in 2 years' time. In New York, it will probably be this year. And in Japan, I imagine it will be next year. So these therapies are about to go to clinic. We have a meeting next week with all the groups in the world to discuss this, as we do every year. And we will be targeting those people we think are going to do well based on what I've told you. What about this group over here? The ones who we think don't do so well? Well, obviously, these are the ones who are very much, I would say, at the forefront for the linked clinical trials, as well as some of these new therapies. Now, those vaccine therapies, which you briefly mentioned, are all being targeted in this group of patients in early stage disease. And there are reasons for that, but I would suspect that it might be more sensible actually to move to that side. And as we know, there are lots of drugs which are now in clinical trial, not necessarily in that group, but these are all trials which have come up recently around that. And we are about to start a trial in the next few months. We're just finishing off the ethics to look at a drug called azathioprine, which is an anti-inflammatory drug in people with Parkinson's disease who we predict are going to do badly on the grounds that we think we can actually slow down and stop them doing badly. That's our trial that we're planning, and that's based on what we've discussed. So, in answer to the question, which was the original question that I noticed on the latest draft, wasn't the question or the title of my talk, but I said, so is Parkinson's disease one disease or several diseases? Who cares? I would say we should all care. I would say, do I think it is more than one disease? I think the answer is no. I think Parkinson's disease is one disease process. You can enter at different points, which is what the genetics tells us. You can come in at different parts of that. What I think is fundamentally different about it, just like cancer, is there is a benign form of this disease where people tend to get it younger, they compensate for it, and they progress very slowly, and they will do fabulously well with the treatments we have now, but we can do better with these new therapies around rescuing dopamine systems, whether it be with growth factors, gene therapies with dopamine in them, or cells. This group have a much more malignant form of the disease. Once it starts, it really progresses quickly. And that group of patients need a whole different type of therapy. And I think this sort of approach should give hope to those type of patients who traditionally are left outside of trials as being not the ones who are suitable, who may be the very ones that we should be thinking about moving forward with some of these disease-modifying therapies. And with that, I will finish. I would like to thank, a lot of people have done lots of work on this, and the great thing is fabulous people support me, fabulous group, lots of terrific international support. And the one thing which has really changed in my research lifetime is the capacity to join up in a way that was never possible before. So people living in their silos doing their own research, that's changed. Genetics changed that. Whatever Hugh thinks, however clever Hugh is, he will only have a few hundred patients, perhaps 1,000 or 2. If you ever want to make a difference, you're going to have to get 10, 20, 30,000. You're going to have to collaborate. And we do exactly the same. As I say, we meet every year to decide how best we can take cell therapies forward. We try and work together with other groups to make sure that what we do links up with what other people do, and that together we can use this type of information to take ourselves forward into a new, better treatment world for Parkinson's disease. Thank
you. Questions for Roger. Claire, I think you're first.
That was a really interesting talk, thank you, and the one before. As somebody with early onset Parkinson's, who is hopefully in your former group, what would you recommend regarding when to join a trial? Because potentially if you go on one trial, you might be excluded for another, and initially I was sold by Professor Morris and the idea of his, and then now yours, and which one should we go for?
It's a very good question, and it's very difficult to answer. So lots of people will say, I'm really interested in joining your trial. I'll just wait for Claire to have the treatment, and then I'll wait, and then I'll see. So lots of people are very keen, but they're always keen for someone else to have it first. And obviously, the first people who enter into a trial, so in our case, the first people who have stem cell-derived dopamine cell, that's a big risk. We think there is no risk, otherwise we wouldn't be doing the trial, but there is an unquantified risk. Secondly, there are lots of guesses we would have to make when we first try it, because we don't know the dose, we don't know quite how to do it. So it's always difficult, because you say, well, actually, I don't really want to be in the first trial. And then you say, yeah, but I've got to wait 2 years to see whether it works. And you say, so it's 2 years to get it going, 2 years to wait to see it happens, a year to report. That's 5 years. Do I really want to wait 5 years? So it's always very difficult to decide when's the best time to join a trial and what's the best trial to do. So I've had someone email actually yesterday saying, which trial would you recommend for my mother to join? And I think it's very difficult. Personally, I always think, what would I do if I was in your situation? So what I would do if I was in your situation, I would make sure that I'm briefed on all the trials. And then I would think, what fundamentally do you think will make the biggest difference to me? Now, I'm obviously biased in what I think. And I think that stem cell derived dopamine cells are going to go big time fairly soon. And the reason I think that is that for years these cells have been out in the wilderness. You know, the idea of cell replacement therapy was all the rage in the 80s and 90s. Deep brain stimulation came along, completely disappeared. And when we started this work in 2006 with cells, nobody was interested. You know, I spent ages trying to persuade people. No one's interested. Well, in the last 3 years, I know of at least $500 million of investment that's gone in by companies to make cell-based products for Pugsies, which have a rational basis around dopamine cells. And I also know that, you know, people are trying to close us down from pharma because they want this space. So that has disadvantages because we're fighting against people who have more money than we do. But it means that people think this has traction, and it means it will happen fast. So my ambition is that when we get funding to do a phase 1 study, the process of doing that will catalyse quick phase 2 studies, and then we will end up with big studies within relatively short periods of time by big pharma with manufactured cells. And in people with young onset disease, I would think that is going to be the way forward, because if you imagine that the main problem of your disease is you've lost 250,000 dopamine cells, well, we can put back 250,000 dopamine cells. That's not actually that difficult. I mean, you may think it's a lot of cells, but given how many cells you've got knocking around in your head, that's relatively small. Now, if you put those cells back, in theory, they will replace the lost cells, and they will replace the need for anything else that's currently available. So think of every drug that's taken. So you won't need to take monoamine oxidase inhibitors, razagiline, sledgiline, because they work on dopamine. Dopamine agonists you won't need. L-Dopa you won't need. You then won't need amantadine because you won't develop dyskinesias because you've got cells instead of drugs. You then don't need deep brain stimulation because that's used for the complications of the L-Dopa treatment you're no longer taking. You won't need duodopa or apomorphine. So suddenly, the treatment with cells, whilst it won't cure you because the disease will carry on, it will have essentially, if it works as well as I would hope, would get rid of the need to take any therapy. Now, you could then say, well, the problem is that's only patching up a disease that's ongoing. Yeah, but those cells, we know the best case in patients with fetal tissue is they work for 25 years. And then the patients died because the patients had them in these trials, you know, in their 50s, 60s. Well, if you've got 25 years for someone to come up with a cure, that buys you quite a bit of time. And I ultimately think that some of these other therapies can then be used with cell-based therapies. So at the moment, trials are done, you have A or you have B. But you could use both. So my ambition would be, you come along to clinic, stick in some dopamine cells. If the vaccines work, you have the vaccine. That stops the protein spreading, if that's to be believed. Thank you very much. Whether we can afford it will be the next thing. I'll worry about that if we get there.
Clive.
I'm absolutely delighted to hear we're talking about using anti-inflammatory agents in Parkinson's. Is there any evidence from stem cell replacement patients so far who've had immunosuppression that this actually makes any difference?
Good question. So first point, just to clarify, they haven't had stem cells. So our trial, which we've completed, is fetal cells. So it's important to get the difference because this is primary fetal tissue. So these are not engineered grain cells. But you're absolutely right. Those patients have been immunosuppressed. Now, it's very difficult to know because they were immunosuppressed for a year and they've had cells put in. And so one of the extraordinary aspects of the cell transplant, there are many extraordinary aspects of cell transplant, but one of the most extraordinary, I think, is there's one patient in particular who had a transplant one side of his brain and did extremely well for 25 years on both sides of his body, even though he had the cells on one side. So the reason for saying that is A, we don't quite understand that, but B, obviously he's the perfect patient to answer this question. Because on one side he's got a cell transplant, on the other he's not, but he's receiving immune therapy. So we can see what's happening on the other side from the transplant, and then on the other side we should say what effect is the immune therapy having on his Parkinson's. The trouble was, because it improved both sides, we couldn't really say. So I think all we can say with the immunosuppression in people who've had transplants is that it certainly didn't make them worse. The patients who've done well have done well, but whether that had anything to do with the immune suppression, we don't know. Some people have tried to look at it the other way around and say, can we look at all people who are on heavy-duty immunosuppression for some reason who have Parkinson's disease. The trouble is the numbers of those patients are rather small, and it's obviously hugely confounded because if you're on heavy duty or major immunosuppression, you've got to have something else wrong with you, and you've got to factor that in, really.
There's one final question at the back, and then I think we probably need to break for
Yeah.
tea because everyone's been sat down for such a long time.
Okay, we have several questions online from people who are listening online, but I'll ask one in particular, which is, are there any early indications of Ambroxol success in non-GBA patients with Parkinson's disease?
That's probably a question for Hugh.
I think the answer is no. I think all they've done with Ambroxol, I mean, my
disagree.
understanding is at the moment.
I've had it.
Have you? And
Yes,
it's cured you?
sorry. I'm non-GBA. I had Ambroxol, and I mean only a short period of time. But I did well on it, and it definitely helped with my tremor. From people who didn't know I was on it, said, "Oh, your tremor doesn't seem to be as bad." And since stopped taking it, my tremor is coming back.
That's the answer.
I know it was a
And
very
then one.
small trial, very small, but.
I mean, my understanding at the moment is the big trial is, the people are trying to work out how much of it gets into your brain and into your CSF, and then plan bigger studies. But I don't know anything more than that.
Exactly.
But it's useful to know that it's well tolerated and had an effect.
So I think with that, it's time to break for a cup of tea. Roger, thank you so much,
Right.
and particularly for hot coffee.